Today I'm going to be talking about cellular respiration, and in particular glycolysis and the formation of acetyl CoA. So glycolysis takes place in the cytoplasm, and it goes through three main phases. The first stage is investment, the second stage cleavage, and the third stage payoff. So through the first stage, we're going to take one glucose molecule and we're going to invest 2 ATP molecules. We're investing 2 ATP molecules because glycolysis is an anaerobic process, therefore it does not use oxygen. ATP is going to act as our activation energy. So basically through this whole process, we just want to break down glucose even further. So first of all, we're going to phosphorylate the 2 ATPs. First, we're going to take one, and we're going to get glucose 6-phosphate. And to show this, glucose is a 6-carbon sugar, so there's 6 carbons. And since we stole one phosphate from ATP, we're going to add it onto the end of this. And next, we're going to use the second ATP and we're going to phosphorylate it, and we're going to make fructose 1,6-bisphosphate. Next, we're going to use the enzyme called aldolase, and basically it's going to end up splitting this molecule into 2 3-carbon molecules, and that's going to be called glyceride 3-phosphate. And then an NAD is going to come through, and it's going to oxidize these 2 molecules right here. And ultimately, it's going to produce an enzyme called GAPDH, and from that enzyme, we're going to steal away 2 hydrogens because there's 2 of these, and we produce 2 of those molecules. So we're going to end up gaining 2 NADHs. And this is important because these are going to be used later on in the cellular respiration process to make more energy. And also in this process, 2 ADPs are going to come by, and they're going to connect with 2 phosphate groups, and we're going to have 2 ATP molecules. And then after this step, we're going to use another enzyme to convert glyceride 3-phosphate into 3-phosphorylate. And with this molecule, we're going to take away a water, and we're going to end up producing phosphoryl pyruvate. And this phosphoryl pyruvate is going to go through a process called substrate level phosphorylation, which basically is going to add on a phosphate group. And when it adds on this phosphate group, another 2 ADPs are going to come by and connect with those phosphate groups and make us 2 more ATPs. And when we produce those 2 ATPs, this is ultimately going to give us 2 pyruvates. Pyruvate is going to make its way through the cytosol, and on its way through the cytosol, it's ultimately try to end up in the mitochondria. But on its way there, it's going to lose a carbon dioxide, and it's going to gain coenzyme A. And since pyruvate is a 3-carbon, it's going to lose one of these, and ultimately going to make us acetyl coenzyme A. And acetyl coenzyme A is going to make its way through the mitochondria, where it's going to be the main molecule for the Krebs cycle. And so just to give you guys an overall view of the glycolysis, we went through 3 steps: investment, cleavage, payoff. Investment is where we use 2 ATPs. Cleavage is when this molecule, glucose, was broken into 2 parts. And payoff phase is when throughout this whole process we have a total of 2 ATPs, 2 NADHs, and 2 acetyl coenzyme A's. And that's pretty much it.